Hi, I'm Lucas and I'm a geneticist. Companies that test DNA are popping up faster than the gray hair on my mid-aged body. While each company aims to provide information about your genetic traits, disease risks, and/or ancestry, they differ in the methods they use, which is very important to consider before getting DNA testing. In this video, I'll compare the three most common methods to test your DNA: genotyping, whole exome sequencing, and whole genome sequencing. I'll give you the pros and cons of each method so that you can decide which is the best for you. First, here's a quick refresher of basic genetics for those that need it, in case you paid more attention to your crush than the lecture in high school. We have roughly 3.2 billion letters of DNA in our genome that are separated into coding and non-coding regions. The coding regions make up a small part of our total genome, between 2 to 3%, while the non-coding regions make up the rest. The coding regions, which are found in genes, contain the instructions to produce RNAs and proteins, which are the workhorses of our cells. This doesn't mean that non-coding regions are useless. These regions contain segments of DNA that are very important, such as protecting our genome and controlling how much RNA and protein is made. As technology advances, it's allowing us to better explore and understand the function and the utility of the rest of our genome. So now that we're all on the same page, let's talk about what these methods are and how they work. For each of these methods, a sample of spit, cheek swab, or blood draw is collected, and the DNA is then extracted. First, let's talk about DNA genotyping, which is a method that looks at hundreds of thousands to millions of letters called SNPs in the genome. SNP stands for single nucleotide polymorphism, a type of genetic variant in which a single nucleotide base in the DNA sequence is changed. SNPs are the most common type of genetic variant found in the genome. Genotyping is most commonly done using a technology called SNP microarrays, or chips. SNP microarrays work by fragmenting the patient's or customer's DNA into segments and attaching fluorescent labels to them. These fragmented and labeled DNA molecules are allowed to react with a specific set of probes, DNA fragments with known sequences, on a DNA chip. If a DNA molecule binds to a probe, it gives off a fluorescent signal and its DNA sequence can be deduced. Next, let's talk about whole exome sequencing, or WES, which is a method that sequences every letter in all the known coding regions in the genome. The process involves fragmenting the DNA and adding adapters. The adapters allow for the DNA molecules to be copied and sequenced. One important step of the WES process is fishing out the regions of interest of the genome, the coding regions, using DNA or RNA probes. The rest of the genome is washed away. The coding parts are then read on a sequencing instrument. Last, let's talk about whole genome sequencing, which reads all 3 billion letters of your genome and your mitochondrial DNA. It uses a similar method to WES, except there is no fishing out regions of interest, since the entire genome is sequenced. On the back end, powerful computers and software are needed to analyze the data and interpret the results. As to be expected, the most computing and interpretation power is needed for whole genome sequencing analysis out of the three methods, given the amount of sequencing data generated. Now let's talk about coverage. SNP microarrays analyze hundreds of thousands to a few million SNPs across the entire genome. For example, 23andMe uses a SNP array that looks at roughly 650,000 letters in your DNA, or 0.02% of your genome. On the other hand, WES reads roughly 2 to 3% of the genome. With WES, each letter in the coding regions is sequenced many times, which is the depth of coverage. The higher the depth of coverage, the more accurate and informative the data. Most laboratories strive for roughly 100x average coverage, meaning that on average each letter is sequenced roughly 100 times. Again, in whole genome sequencing, all 3 billion letters are sequenced, and here again the depth of coverage matters. Most companies provide a roughly 30x average coverage. Now on to the cost. The cost of all three technologies is dropping rapidly, but currently many commercial labs are charging roughly $100 plus for genotyping, $300 plus for whole exome sequencing, and $500 plus for whole genome sequencing. These prices include sample preparation, processing, sequencing, and interpretation, but vary significantly, mostly depending on the depth of coverage or number of SNPs analyzed and amount of interpretation reports provided. It's also worth noting that since our understanding of genomics is constantly advancing, many labs have transitioned to a membership fee model on top of the initial price to offset the cost of data interpretation and generation of reports. So let's take a deeper look at what these three genetic testing methods can tell us. While DNA genotyping does only cover a small amount of the genome, the chips do contain many SNPs associated with traits, health, and for ancestry purposes. This type of testing is great for providing information quickly on common genetic variants that increase your disease risk, make you unique, and give you clues about your heritage. Where this type of testing is lacking is obviously coverage of the rest of the genome, especially the coding parts. This means that if you want to know if you have a detrimental variant outside the SNPs covered or additional information about your traits, health and fitness, you're better off getting whole exome sequencing or whole genome sequencing. For example, if you want to know if you have a genetic variant in the BRCA1 gene that increases your risk of breast and ovarian cancers, you'll be better off sequencing the whole gene because DNA genotyping only looks at a handful of nucleotides in BRCA1. As previously mentioned, whole exome sequencing does cover all the known coding regions of the genome. The good news is that it is estimated that roughly 85% of the genetic variants associated with health and disease occur in the coding regions. The bad news is that we're starting to understand the functional importance of variants in non-coding regions and their important roles in human health at a rapid pace. This means that West and genotyping data is increasingly becoming obsolete. For instance, more disease-causing variants in BRCA1 and other genes are being identified in non-coding regions, especially in promoter regions, which are upstream of the gene, and in introns, the DNA segments in genes between exons. If you want to see the whole picture in terms of your genome and minimize future genetic testing, then whole genome sequencing is the way to go. WGS improves on West in that it also sequences the remaining 98% of your genome. There is still much unknown about the non-coding regions of your genome. Discoveries are coming at a rapid pace. Equally important, there are many types of variants that impact our genome aside from SNPs, such as structural variants, which are large fragments of DNA that are rearranged, and copy number variants, which are large sequences that are deleted or duplicated. WGS enables exploration of more of these and other types of variants across the genome compared to West and DNA genotyping. So to summarize, the main advantage of genotyping is its affordability and speed compared to the other sequencing method. However, it is limited in the amount of information it can provide as it only examines a small portion of the genome. West is more comprehensive than genotyping, providing information about more mutations and variations that can impact health, wellness, and disease. One of its limitations is that it only sequences 2 to 3% of the total genome, meaning that much genomic information will be missed. WGS has the ability to identify both coding and non-coding genetic variations. WGS has a higher cost and longer processing time compared to genotyping and West, making it less accessible to some individuals. However, its comprehensive nature provides the most complete picture of an individual's genetic makeup, making it very useful for personalized medicine, especially as our knowledge base increases. My final thought, given the current cost of these methods, I think WGS will give you the best bang for your buck.